Dear grade 12s, welcome back to another video lecture. This video lecture is going to cover the topic of meiosis. Previous video lectures we discussed in depth DNA, the code of life. So if you haven't watched those videos, I urge you to go and watch those 3 video lectures because that is going to help you understand the topic of meiosis. And we obviously know, based on previous grades, that meiosis is the division of sex cells, the division of gametes. Whereas there's another type of division which is known as mitosis, and we don't want to confuse the two. One is mitosis, one is meiosis, and mitosis deals with the division of somatic cells or body cells. And with that being said, let's get into it. Before we get into the grade 12 content, I think it is important that we first understand a little bit of previous knowledge, especially from grade 10. When we did DNA, we discussed chromosomes, we discussed genes, we discussed DNA molecules, and I showed you in previous videos where each term comes from and how all these terms are just smaller subsections of one another. But a chromosome, in simple terms, is a DNA molecule that is coiled around histone proteins. And based on previous video lectures, we know that histone proteins are there to keep the structural integrity of the DNA molecule. And it is also there to make sure, with the structural integrity, it is there to make sure that this DNA molecule does not break or it doesn't become injured during any metabolic processes. This one here, this is how it looks. If you microscopically, if you zoom into it, you'll see it being coiled around some proteins, but simply it just looks like that, which we know it to be more commonly, we know it like that. And this is an example of an unreplicated chromosome, but based on 2024 NSC marking, we don't use the word unreplicated anymore. For both unreplicated and replicated chromosomes, we can just use the term chromosome for both, and they will mark you correct. So don't use unreplicated and replicated. They do not accept that anymore. But I put it in here to just show you that they don't accept it anymore, and that obviously also what is accepted and what is not accepted. Moving on, we obviously know about chromatin network. When we look at the structure of the nucleus, you have your nucleolus, your nucleoplasm, your nucleopores, but inside there is this thread-like network which is known as the chromatin network, and chromosomes appear in the form of chromatin network when the cell is not dividing. But when the cell is starting to prepare for division, those chromatin network, or those jumbled up chromatin are going to thicken and shorten, and it's going to form a single chromosome. You see, note I didn't use unreplicated. Single chromosome is fine. It's now a single chromosome. But take note, this is how it looks before division. This is before division. After division, it looks like that. And it's understandable because division, before division, that's before division, and before division is replication. So basically before division and replication, I forgot to include there, and replication. So basically before division and replication, it looks like that. But once replication takes place, it is going to look like this. Remember, replication, you are just making another one. So obviously there's going to be 2 now, right? That is replication. And after replication, division takes place. So in order for division to take place, replication must also first take place in order for division to take place. And then based on that, we know that that is a chromatid and that is a centromere. Those are previous concepts that we do know from previous grades. But to get into the grade 12 content. Now, with the grade 12 content, as I discussed earlier, we have somatic cells and we have gametes. Now, your somatic cells are body cells, and this here would go hand in hand with mitosis. Gametes and sex cells will go hand in hand with meiosis. which is what we want to cover. But just a quick revision on mitosis. We know mitosis is broken up into PMAT, and obviously, in the prophase, we have the disappearing of the nuclear membrane, we have the disappearing of the nucleoplasm, we have the chromosomes in the nucleus, right? And then we know the nucleoplasm goes to a jelly-like fluid because it needs to keep those chromosomes within the cell, right? We also know that the nucleolus over there disappears, and the chromatin network it thickens and it shortens, and it goes through all of those processes. That is in your prophase, obviously. Then, obviously, when we come to your metaphase, and obviously we also know there that spindle fibers form, right? And also now, obviously, in the metaphase, we know that they go and all lie on the equator. This is just a brief summary. They all go and lie on the equator. Very important. The centromere of the chromosome is always attached to the spindle fiber, right? They go and lie there at the equator. That is your metaphase. Then in your anaphase, we know that it is pulled right to opposite sides. In your anaphase, it would be pulled to opposite poles. So if there was two, one would go that way, the other one would be more this way, as it is being pulled to opposite poles. Then we also know when you come into the telophase, before you get the telophase or in the telophase, something happens and sort of looks like Pac-Man. Oh, my drawing is always, but let's just do it like that. There's going to be some invaginations over there. Do it nice and big to show you that there's going to be some invaginations, right, where the cytoplasm is dividing, right, and when that cytoplasm divides right through, there's going to be a chromosome on the side, and there's going to be another chromosome on that side, and that is how we went from 1 to 2, right? And then after the end, you're gonna have 1, and then you're gonna have another one. So you went from one mother cell to two daughter cells. That is just mitosis in a quick summary. That's not everything that happens there. Obviously, you can go and look at what happens there in your own revision, but for the sake of keeping this video based on the grade 12 content only, that was just a quick recap on mitosis, just so you can understand. Understanding mitosis will really help with understanding meiosis. With that being said, let's just go to somatic cells. Now, somatic cells, now this is a very important concept that we need to understand. This concept over here is a very important concept that we need to understand. That if an organism reproduces sexually, the chromosome number is always even. Because humans reproduce sexually, they will always have 46 chromosomes. In certain cases, they will never have 47 or 48. They will have an extra one on a pair, right? But they will never have 47, because if you have 47, then you're a completely different organism, right? Because a human has 46 chromosomes, a potato plant has 48. So if you go from 46 to 48, you might turn into a potato, right? So that is a very important concept we need to understand here, that if any organism that reproduces sexually by sexual means will have an even chromosome number, right? Dogs have even, cats have, mosquitoes, birds all have even chromosomal numbers, right? Things that won't have chromosomal numbers are things, for example, like amoeba, because of binary fission, because that is asexual reproduction. But if it's sexually reproduced, it will have an even number of chromosomes, right? And when we say even, it means that it will always have a double. What do we mean when we say double? It means that this, when it's broken up, will become two odd numbers. And this doubling effect, right? This way, two odd numbers or two separate sets of chromosomes give us one set of chromosomes is when it's like that, when it is even with double set of chromosomes, it is known as diploid, right? It is known as diploid. So this here is a diploid chromosome cell. It's a diploid because it is even, it is double, right? It took two to make one. That is diploid, right? And what is very important to also understand about chromosomes is that a set of chromosomes, 23 comes from your mother, which is obviously your maternal set of chromosomes, and 23 comes from your father, and that is your paternal chromosomes, right? And based on the strength of the chromosomes and the strength of the genetics of your mother and your father, you're going to end up looking more like your mom. That means that your mom had stronger genetics. If you look more like your father, it means that your father has stronger genetics. But you will have certain characteristics from your father and certain characteristics from your mother. For example, I look like my mother, but I'm getting early gray, which is a characteristic I get from my father. So you get certain characteristics from your mother and you get certain characteristics from your father. Alright, so 23 comes from your mother, 23 comes from your father, and together, so that's 23, will give you your 46 chromosomes, right? In the human cell. And that's what I noted here. You get your paternal chromosome and your maternal chromosome, your maternal and your paternal. And your maternal chromosome from your mother and your paternal chromosome from your father together, they are called a homologous chromosome pair. A homologous chromosome pair. That's a very important term that we need to remember when we get into meiosis 1 and meiosis 2, which will be in the next video. But they are known as a homologous chromosome or chromosomal pair. They are paired, right? Because for every maternal chromosome, there will be a paternal chromosome, right? For every mother, there has to have been a father, or there has to have been some sort of spermatozoa with the egg. So that's the understanding that we have here. For every maternal chromosome, there is a paternal chromosome because we are reproduced sexually, which means there will be a male and there will be a female involved, right? Even if it's done through technological means. Now, that was for somatic cells, right? So somatic cells will always have 46. Very important to understand. Somatic cells will all have, somatic cells never have 23 because that's where meiosis comes in. Because meiosis is known as reduction division, which means it has a halving effect. So that halving effect to form 23 and 23, that is where meiosis comes in, the halving effect. So don't get it confused. Mitosis has to do with 46. Meiosis has to do with 23. The 23 and the 23 fusing together to give you the 46. And when you do human reproduction, you will see that when I do it, go over it, you will see that in more detail. But gametes, remember this was a set from your mother and a set from your father, right? So that means that this is one set and that is one set, right? And we're going to look at N and 2N and then I'm going to show you. So with gametes, and obviously gametes here deals severely with meiosis, right? Will deal severely with meiosis. So gametes only have one set of chromosomes. What do we mean when we say one set? They have 23, right? Because you must remember the two gametes are the egg and the sperm, and the egg and the sperm will fuse. So 23 and 23 will fuse to give you 46. And therefore we call them haploid. So the difference between diploid and haploid is haploid on the one hand is half, right? Haploid is half and diploid is double. That is a very important understanding that we must grasp here, right? Haploid is half, diploid is double, right? So if you start with 46 in diploid, it means that in haploid you must have 23. If you start with 4, right? In diploid. In haploid, it means you must have 2. It's a halving effect, right? Meiosis is a reduction division, meaning it has the halving effect, right? And this is a very important note. Haploid is half of diploid. Very important. Haploid is half of diploid. So in humans, you have 2 sets of haploid. That's why we call it N and 2N. So you have N equal 23 and N equal 23 from your mother and from your father. And when fertilization happens, okay, I did actually write it here. When fertilization happens, right, with the egg will have 23 and the sperm will have 23. When fertilization happens and the acrosome of the sperm and all of those stuff, it breaks the layers and all of that stuff when we look at human reproduction. N and N fuse to give you 2, and 23 and 23 give you 46 to form that zygote. And you know that is normally say about 2 to 4 days after sexual intercourse is when the zygote forms. Then that zygote is going to undergo mitosis. And the reason it undergoes mitosis is because we don't want any more reduction division. We don't want a halving effect. We want a doubling effect because we want that zygote to grow. So in order for it to grow, it needs to make more. And by making more, it grows. And then with all of this making more, there's all of this mitosis, it forms an embryo with a lot of cells having 46 chromosomes, right? From that is how you grow. From 1 until your age, whatever the age is now, you have gone through processes of mitosis. As you're sitting and doing something, mitosis is happening constantly to repair the old repair the old cells, right? Or to replace the old cells, if I can even say that, to replace the old cells to make sure that the new cells are formed, right? To make sure that the new cells are formed. So growth is heavily due to mitosis, right? The repairing of cells is heavily due to mitosis. When you have a scab, when the skin forms over that scab again, that is mitosis taking place because skin cells are then being formed over that scab wound. Right, so very important to understand here. Haploid is half of diploid, and in humans you get 23 from your mother, 23 chromosomes from your mother, 23 from your father. Together they fuse to give you the 46 chromosomes, right? To give you the 46 chromosomes, right? And then this here we call 23 pairs because chromosomes occur in pairs, obviously, right? It will occur in pairs, and when we look at the karyotype now, you'll understand what I mean. So how do we know how much chromosomes humans have? This is when the karyotype came. Now the karyotype shows a complete set of chromosomes in the cell of an organism. The complete set, so everything. That is one definition that can be used, and remember this can come in biological terms. In the paper, it can come in biological terms, it can come in multiple choice questions, it can even come as a definition, right? So make sure that you know it. And then another one, obviously the same thing applies here. It's a representation of the number, shape, and arrangement of the chromosomes in the nucleus of a somatic cell. Now, why is the karyotype so important? And we're going to look at it now. We're going to look at it not in this video, but in a later video. We're going to look at something called non-disjunction. And non-disjunction explains why a child has Down syndrome. A Down syndrome is a chromosomal mutation. And this karyotype shows us where the chromosomal mutation occurs. And I'm not going to speak too much about that. That will be a separate video. But in the human karyotype, you have 22 pairs of autosomes and 1 pair of gonosomes, or 44 autosomes and 2 gonosomes. Right? Remember, if it occurs in pairs, right? So there's 22 pairs, which means there's 46 of them. Right? Individually, there would be 1, 2, 3, 4, all the way on to 44. There are 1 pair. 1 pair of gonosomes means that there's 1, 2, so there would be 2 gonosomes. Now, the differentiation here comes in between gonosomes and autosomes is that Autosomes are not involved in sex determination. However, gonosomes are involved in sex determination. What does this mean? Autosomes are not involved in determining whether the child is going to be a boy or a girl. Gonosomes are involved in that process. Gonosomes on the karyotype, if you look at the karyotype in the exam, so gonosomes would also obviously mean that it's the 23rd pair, right? It is the last block on the karyotype. Those gonosomes, those two gonosomes, are going to tell you whether the karyotype is depicting the cell of a girl or the cell of a boy. And from that you'll be able to tell if it's a girl having, if it's a girl that has Down syndrome or a boy that has Down syndrome, right? Or various other syndromes that or affect, but I'll do a separate video on all those syndromes. But let's just quickly talk about sex determination. So XX, if on a karyotype you see the 44 pairs, the 44 autosomes at XX, that means it's a female. Automatically, it will be a female. If you see XY plus 44, it's a male. If there is a presence of a Y, right, it is a male. Always a male. Even that is why I said here, there's even in this case here where you have XXY, because there is a Y, this will be classified as the karyotype of a male, the representation of a male karyotype, because of the presence of a Y. Even though there's two X's, those X's just means that this male will have severe female characteristics. This male will have full on breasts, full on developed breasts. The male will have female genitalia. The male will have a lot of female characteristics, right? And because of the XX being present, the male will have reduced testes, and therefore the male will be infertile, right? So just take note, if there is a Y present, automatically it is a male. And if I show you on a karyotype, if I show you the male, so up, so from here until here, that is all your autosomes. And if we count it, can we see that there's 22 pairs? But if you count it individually, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, all the way, you will count 44 autosomes, or 22 pairs of it. 22 pairs of autosomes. Now, how do we know that this is a male? It is because of this, and they circled it so nicely from where I got it, right? The big one here will always, so we have a big one and we have a small one. The big one will always represent the X. The small one will always represent the Y. Please keep that in mind. In exams, if they give you a big one and a small one, that is how you differentiate between whether it is male and female. The big one is the X. Remember, this is the 23rd pair. The big one is the X. The small one is the Y. So because it's a big one and a small one, this is a male. Right? Even if, for example, now the odd case that I showed you, where it will have a big one, and a small one. Right? A big one, and a small one. X, remember the big ones are always XY. This is still a male because of the presence of a Y. Please keep that in mind. Then with a female looks similar. From up until there, it looks the same. Right? Pardon me, it looks similar. You will have 44 autosomes, or 22 pairs of autosomes, right? And this XX here tells us what? It's a female. Remember, now we can see that there is two big ones. That is XX. And that tells us that this is a female. Right? That will tell us that this is a female. So, in summary, on a karyotype, if the presence, if there's a Y, that will always be a male. XY is a male and XX is a female. even if you have XXY, that is still a male but will have a lot of female characteristics. If you have XYY, it is still a male, but now this male or this male has a lot of male characteristics, and they are normally psychopaths. Someone that has XYY is normally most likely going to become a psychopath because they have an added level of male characteristics, which means that they have an added level of aggression. And that normally makes them psychopaths. And it's scientifically proven that most murderers, most psychopaths, most um, sociopaths, serial killers, all of those types of things add an extra Y on the 23rd day because that Y, remember the Y means the presence of a male. Now, if a normal male, okay, let me not generalize, shame. Not all males are sociopaths or psychopaths. But if a normal male has a certain level of aggression and it's just XY, you can only imagine that if another Y is added, that level of aggression will increase so significantly that the likelihood of that male becoming a serial killer or aggressor or whatever is so it is just 100 times more the likelihood of that happening, right? And then also I just want to quickly touch on something. The egg cell does also not determine whether it is a girl, right? The egg cell does not determine whether it is a girl, right? A girl or a boy. Because the sperm, so remember the egg will have the X, but it is the sperm cell that determines whether it is the father or the male that determines whether it is going to be a girl or a boy. Because sperm cells are either X or Y. So if X fuse with X, if the egg, remember the egg is always X because it's female. But if a sperm comes along with the X, that means it's going to be XX, which means you're going to get a female. But if the sperm with the Y comes along and fuses, it means you're going to get a male. Right? So understand the difference. The egg cell does not determine here whether it's going to be a girl or a boy. It is the sperm cell that comes along and will determine whether it is a girl or a boy, because sperm cells occur in X and Y, and the X cell only occurs in X. In the next video, I will do meiosis 1 and meiosis 2, and then a couple of examination questions on meiosis, the ones that are interesting, and 2024's one, which was also quite an interesting question as well.